In our surroundings, there are some tiny little things that can cause diseases, like influenza when they get into our body. What are they? They are viruses. Viruses are extremely small non-cellular particles. Most of them can only be observed under an electron microscope. They consist of nucleic acid, either DNA or RNA, surrounded by a protein coat. Some have an additional outer membrane, called an envelope. For example, hepatitis A virus is a non-enveloped virus, and influenza virus is an enveloped virus. On the protein coat or envelope, there are surface proteins. These proteins can recognize and attach to specific cell types of their hosts. Viruses exist in a variety of shapes. For example, a coronavirus looks like a ball, and a bacteriophage looks like an octopus. Viruses must infect a living host cell to multiply, because they lack organelles and enzymes for metabolism. The infection is often specific. For example, hepatitis B viruses infect only human liver cells, but not other types of cells. Multiplication of viruses involves 5 major steps. A virus first recognizes and attaches to a host cell by binding its surface proteins to specific receptors on the host cell. The viral nucleic acid enters the host cell. The nucleic acid directs the machinery of the host cell to synthesize new viral nucleic acids and proteins. Viral nucleic acids and proteins then assemble into viruses. The newly formed viruses finally burst the host cell, and they are released. They then spread to infect nearby host cells. However, some viruses may stay inactive after entering the cells. They enter a latent period. The viral nucleic acid is integrated into the DNA of the host cell. It is replicated along with the DNA of the host cell when the cell divides. No viruses are produced, and the host cell can function normally. However, the virus may be reactivated at any time. Once it is reactivated, it produces new viruses, which spread to infect nearby host cells.